All right, good morning. Good morning. I found my focus mode button so you can be on camera again. I get to see your lovely faces. All right, we are going to do some practice work today, but I do have 3 people who need to finish the benchmark. Malaysia, you are one who needs to start, I believe. And where's Andre? He needs to finish. And who else do I get? Avane is not here yet. Okay. All right, give me a minute because I have to ask Mr. Mann to open that. And then I'm going to work. Then those of you who are testing, I'm going to ask you to mute so that we can work with everybody else. And there they are. Okay. All right, so while I'm waiting for a response from him, I want to go over 2 messages that are important. All right, Andre Abernay, good morning, good morning. I need you to finish the benchmark, but don't hold on for a minute because Mr. Phillips has to open it. They keep telling me that. Okay, sorry. I have been on hold with these people for over. You finished the benchmark? Okay. All right, last time I checked, you had not. So it's just Malaysia and Andre then. Take that off of there. Why is it not sharing like I asked it to? Asked it to share. All right. So again, you have to view this as a student. Actually, as a student, I couldn't click on the reply. All right. So make sure you check the announcement. All right. Announcements. Let's see if it goes like that. All right. Here we go. Can I open it? Hey, must do before spring break. All right. Again, you're using your own laptop when you go for the milestone, not just biology, but all the milestones that you have to do. So, and you go to a testing site. So you do this in person. Okay, some of you didn't know you actually have to go to a test site to do this, and you have to bring your laptop. They want to make sure that your Chromebook is working because in the middle of the test, you don't want anything to go wrong. If it's not working, they may not have another one available for you. So it's important that you check. So there's a little link here that you can go through and make sure that you go through all of the steps to make sure that everything on your laptop is working. There shouldn't be any damage, no cracked screen, no keys not working, and your sound has to be working for your English test. So make sure that you check that out. Please let Ms. Card know if you have an issue with your Chromebook before the test. You can't wait 2 days before or the day of and say, oops, it's not working. So please, well, I guess if it's not working that day, you can't help that. But if you know that it's not working now, please let them know and they'll try to swap it out for you or get it fixed somehow. All right. That being said, let me see. Let me see. All right. Mr. Phillips said he opened the biology test. So those of you who need to finish it, and it does need to be finished. Notice it has to be opened and closed every time we do this, and we don't want to keep doing that. So please focus on getting that finished today. If you need to get the benchmark finished, Andre, Malaysia, that's you. Please go log into Progress Learning. You're looking for the LVA benchmark number 3, and please take care of that. All right. Whoops. That being said, honest to goodness. All right, let me get back to where I'm supposed to be. Okay. That wasn't for you to be watching. Okay. On your home page, for everybody else, and then those of you who are testing, you need to listen to, on your home page, you have something called Biology Milestone Test Practice. Please click on that. Click on that. All right, so what we're going to do for the rest of the week is do these milestone practice tests. So once you get there, let me do my screen big so that I know you're looking at what I'm looking at. All right, so and I need everybody to go there. I'm gonna jump over here can I see what you're doing? I see a lot of blank screens. I need everybody to click on the link and then move with me because we're gonna go through these questions. I don't know whether you are or you aren't. All right, there we go. Renee, are you with us? All right, Andre, I need you to do the test. So please log into the test. All right. Everybody, please log on to the link that I just put up there. All right, so I'll back up again. This is where you should be, and that you can find that on your home page. I need you to do progress learning first, Andre. Okay, Avane, this is not on your home page right here. It should say biology milestone test. It's not going to be in the module. I think it is in your module. Let me check. You're on it? Okay. But I do believe it's in the modules, just in a funny little place. And the reason it might not show up the way you think it should show up is, all right, so it's under biology EOC test practice. So it's the milestone test practice. So it is in the module. It's in its own little module. All right, so if you click on that, it'll take you here. And we're going to click on the little green button. And we're going to click on online testing tools. And we're going to click on EOC. And we want study guide. No, we don't want that one. We want test practice today. We're going to do this one later. Tomorrow we're going to do this one, but today we're going to do this one. Test practice. Standard online tool, biology. All right, so this is a sample of what the test actually looks like. So when you go to the test, you'll get a label with your name on it and the session number and tell you what test you're taking. There'll be a proctor that will walk you through this. And one of the things they're going to ask you to do is make sure that your name shows up the way it's supposed to show up, because if not, the scores will not go where they need to go. And you'll hit continue, and they'll keep on reading those instructions to you. We're going to do a practice test. Okay, hold on, I have to take this. know how many days he'll leave service? Okay, thank you. What your name again was? John. Sorry about that. Okay. Hopefully you, so again, at the test site, the proctor will read all of that for you, and we'll go through all of these introductory things. And so there's a lot of information, and I, y'all have done tests before, so you know there's a lot of information that they read through before you begin the test, and we're going to begin the test. Oops, begin the test. All right. So, what we're going to do today is just go through these questions. And again, what I want you to understand too, in fact, um, take a minute to look at this question. I'm going to stop sharing for a minute, do attendance, and come right back. And then I won't forget. Be right back. Attendance with you guys is always easy because everybody comes to class. All right, Andre, you should be in the progress learning. Malaysia, you good? You want to do the biology, LVA biology. Yeah, that's the one you want. There you go. Okay, so you're good. Andre, we need you to finish. We need you to finish. So please focus on that first. All right, let me do attendance. Navane, you sure you finished? Last time I checked, I didn't see a score. Everybody's here. Okay. All right, back to our question. All right, so these test questions, are samples now. You're not, one of the things I don't like about going over review is that people say, oh, the questions that we went over, I didn't see that on the test. You're not going to see that on the test. What you're going to see is questions like this on topics like this. So as we go through this, just know the kind of format that they use, because they're not a question um, that you're going to be used to seeing. So you need to, this is to get you used to the way they ask questions. And so yes, you see with this one, it's a scenario and you've got to examine the scenario. Now, this was an area that most of you kind of struggled with when we did the phases of mitosis and meiosis. You had kind of, so go back and study those, okay, so that you know PMAT, when is prophase, anaphase, metaphase, telophase, and the final stage where it divides in two, okay? All right. So the picture they have, you were in the classroom, you might have a paper plate, and they use some popsicle sticks and Q-tips to represent the um, chromosomes. And you need to examine every single picture before you try to answer the questions. Okay? So remember that. And then vocabulary, know your vocabulary. So we need to look at this and say that we're looking at homologous pairs. That means they're the same. So there's two Q-tips. That's a homologous pair. And the two popsicle sticks, that's another homologous pair. All right. So in this particular example, you have 4 chromosomes, 2 pairs. All right. And then they're going through meiosis, and we end up with 2 identical daughter cells. Question says, which model explains mitosis, how it maintains genetic continuity? Genetic continuity means that what the genes we start with, we're going to end up with those genes at the end. So that's continuity. So you got to pay attention to the phrasing of things, and I'll show you why later. All right, so in this one, the chromosomes in cell 1 are the same as 6 and 7. So in cell 1, we have what the plate represents the whole cell. This little circle on the inside represents the nucleus. And we have our 2 chromosomes inside. After the split, again, 6 and 7 show 2 chromosomes surrounded by a nucleus, and there's 2 of them. So 6, and so number 1 is the correct answer for, or A is the correct answer for this. This one, number 2, the nuclear membrane has disappeared and they're starting to move away from each other. This would be metaphase, where they're lined up in the middle. This
You're
one,
not. I don't think you're.
oh,
Are you supposed to be sharing? You're not sharing.
yeah, I am. And I click share, and thank you. I'm doing all that talking. And well, hopefully y'all are on that page too, so you were still following me. All righty. We got it. Okay, there we go. Thank you, Imani. You always keep me on my toes. All right, let me bring my screen up, and that way I can tell with them. Okay, here we go. All right, so again, in the first one, there's a nucleus around the Q-tips and popsicle sticks, or our chromosomes. In the second one, there's no nuclear membrane. They've started to move away from each other. They're lined up in the middle in number 3. Number 4, our homologous pairs have been pulled apart, and they're moving to the poles. And then in 5, a nuclear membrane is starting to form around our homologous pairs. And then the cell can divide. And once it divides, then they duplicate again. And 6 and 7 look exactly like number 1. Now, so A is the correct answer. Crossing over is a word that you need to know. You need to know during which phase. Cell number 4, it says occurs in cell number 4. And that's not, and it does increase genetic diversity, but it does not occur in stage 4. So you need to go back and look up what is crossing over and during which phase of mitosis it occurs. So again, this is a question that they're asking you, but they could have asked you about crossing over. So you need to know what that is. You need to know when the nuclear membrane reforms in 5, each one has twice. And right now that's not true. It doesn't have twice yet. Okay. You still have 2 homologous pairs. They have not duplicated yet. So there's not twice the number of chromosomes as cell 1. Okay. So read each one carefully, because sometimes the difference is so slight. You need to know what independent assortment means. Let me go back up here. Genetic diversity. Yes, genetic diversity means we get differences and everybody's not exactly alike because of crossing over. A little piece breaks off and jumps on the other's chromosome, and then it creates a slightly different chromosome than you started with. So that's genetic diversity. Independent assortment, again, means that sometimes one goes one way and goes another way. So vocabulary words, spend time with your vocabulary words, because if you know what the question is asking, then you know how to answer it. Okay, you know what your words are, you know how to answer it. I'm a full believer in studying vocabulary. All right, this question. Any questions on that one? All right. This question has to do with our macromolecules. So that's something we did way at the beginning of the year. We have a graphic organizer with that, so find that and study that because they ask a lot of questions about graphic organizers. So we looked at the structure. One of your columns was structure, and based on the structure of this first one, that it is rings of carbon bonded together with hydrogen and oxygen. That's a description of a carbohydrate. So now we need to know what is the function of a carbohydrate. Okay, carbohydrate gives us energy and it gives us short-term energy. Okay, so if you want quick energy, you eat a carbohydrate. Okay, next one says one molecule of glycerol and connected to 3 fatty acids in chains. And there's a lot of carbon and a lot of hydrogen. That describes a lipid. Function of a lipid is also energy, but you get a whole lot more energy from lipids than you do carbohydrates. Okay, so that would satisfy this question. Catalyzing biochemical reactions are enzymes, and I believe you have enzymes on your macromolecule chart as well. And storing and transmitting genetic information, that's your DNA. And that is also on that macromolecule organizer that you made. So again, pull that out, study it. All right, so that's how you would answer that question. Anybody got a question on that one? All right, this is a question we have not covered yet, but we are going to cover it after the spring break. So spring break starts Friday. You'll be back on the 6th. And then we have 2 weeks to kind of do a quick crash course in ecology and evolution and taxonomy. Those are the 3 things that are left. So we're just going to touch on the important parts of those, but we're going to keep on practicing for the test. The link is available to you. So over the break, you'll be expected to look at this because we want to make sure your score is the very best they can be. All right, so here the vocabulary word here is invasive species. Okay, invasive species means that they are not native to the area. They come from somewhere else and have taken over. So they have invaded the area that they're been introduced to. And most of the time they cause damage because what they do is they compete for the resources that are there. They outcompete the organisms that are native to that area. So those natives either die or they move away. All right, so some insects bore into trees and then they get transported when the trees are cut down in numerous products. Wood crates used for packing materials may be infested with insects or eggs that are transported over long distances. What is one safe, practical way that people can act responsibly to prevent the spreading of potential harmful invasive insects? All right, now they give you another thing about reading paragraphs is they give you a lot of information, but that's not necessarily because none of the questions have anything to do with crates, but it is in the wood. Okay, so the first one says store unused firewood from one winter until the next winter in a shaded grassy area. Well, that just gives the insects time to, because they're going to feed on the tree. Um, they're going to have a field day with where they are. So we don't want that one. Spray all wooden products with pesticides before bringing them into your house or the building to kill the insects before they enter your structure. That sounds nice, but you're spraying the product that you have inside the crate, so that's not a good idea. All right, chop down trees suspected of being infected and use a wood chipper to make mulch and landscape. And that's not going to help you because it said prevent spreading. That's not one. This last one is your best one. It says buy local firewood near a campsite. So that means if you go camping and you need fires, don't bring it from home, bring it to the camp. Buy whatever's they have available instead of transporting wood from another area. And then if you have extra, don't say, I paid for this, I'm going to take it home. Leave the extra there, because then you would be transporting that infestation home with you. Okay, so that would be the best one. Okay, you don't want to, this one you're killing the tree and saying that I'm going to kill it off so the bugs don't get it. You don't want to do that. Cut down a tree unless you have to. So this is the best choice, that you buy it locally, leave the wood where you got it from, and then leave any extra for somebody else to use, and don't try to take it home with you. Okay, so like I said, we're going to look at ecology. This is in the ecology unit. I believe I have published all the, let me go look. Hold on. If I haven't, let me do so right now. All right, modules. And I say this because you can, over the break, if you want to look ahead, you can look at these also. So yes, ecology is published and evolution is published. So you should look at those. Is this published? No, it's not published. It's kind of is, but kind of, it's not published. So I am going to publish that for you so that you look at all of this. And there's taxonomy. So there's three things that we have to kind of brush over before you do the test. All right, this one should look familiar. This is genetics. And then here you need to know the role of the 3 types of RNA. And here again, you also should have a graphic organizer so that you know what each type of RNA does. And it's important that you understand the picture, because some of these questions, some of these answers are kind of good, but they don't relate to the picture. And they give you a picture for a reason. So study the picture. What this picture is showing you is that this little tRNA that they've labeled is attached to an amino acid. And the arrow indicates that it is bringing it to the ribosome. This structure represents the ribosome. This is kind of like the little conveyor belt, because ribosomes are where we make proteins, and we make proteins with amino acids. All right. So looking at that picture, the best answer is going to be the first one. It tells you that the tRNA delivers amino acids to the ribosome. That's what it does, and that's what it's doing in this picture, so that they can be added to the developing peptide. Again, vocabulary. Know that the protein chain is called a peptide chain. So it's going to bring the amino acid to, its job is just to bring the amino acid there. Then they connect to each other, and the transfer RNA goes away and goes and gets another amino acid and comes back until they finish making the chain. All right. tRNA recognizes the stop codon of developing peptide so that no new amino acids are added. That is a true statement, but that's not what's happening here. This one is adding one. So you have to look at the fact that it says in what the role is in the process shown in this model. So again, look at the picture. Like I said, this is a true statement, but that's not what the picture is showing you. Okay. And remember, here's another vocabulary: stop codon, stop and start. It knows when to stop making that chain of protein. So you notice this one has nothing in it. That was a start codon, and then it'll be blank again when it stops. So again, that's a true statement, but not true for this picture. tRNA signals the release of the peptide from the ribosome once all the amino acids are added. That is not a true statement. That's not its job. All right, tRNA scans the developing peptide to make sure the sequence is completed. That's also not its job. So the first 2 are true, but the first one describes what's happening in the process shown in this model. Okay, again, take your time, read the question thoroughly. Okay, because they're trying to trick you. And my test that I give you, all the answers are obvious, but they're trying to trick you. So I want you to think about it. All right, this one we did bacteria a little bit, and again, when we talked about cells and genetic propagation. So the diagram represents a model of how bacteria become resistant to antibiotics and allow bacteria to survive treatment, which best explains what's indicated, how the indicated step in the model allows the bacteria to develop resistance. All right, this one is what it starts with, okay? And then it develops, some of them develop, and then you got these ones with the X's on them, which are not the same as those over there. So somewhere in here, they've mutated, right? So that over here, the antibiotic is killing all the ones with the circles, and the ones with the X's have survived. And at the end, only the ones with X's inside are generated. So the antibiotic only worked on the ones with the circle. So initially, that's what they treated for the one that they saw, but then in this space, the genetic mutations promote resistance because you you so it multiplied. So even though it had a chance to multiply, it also multiplied these guys that are resistant. Okay, so when you get over here, the ones with the circle have died. So okay, they mutated in this step. Again, it's got to be in by the indicated step in the model. So you got to study the picture. They put a box around this step. That's the part of the question you need to be focused on. A portion of the DNA image is re-replicated. Okay. All right, let me come down. All right, so it has nothing to do with alleles in the antibiotic. So that one's wrong. All right, moving on. All right, Punnett square. This was another area that some of y'all struggled with. But notice they're going to give you a key. The thing here is to understand your vocabulary again. When they give you the capital letters, those are dominant. So they're telling you, then they haven't said that, but they're showing you capital R is red flowers. That means red flowers are dominant. Then they're showing you the little r's and they're telling you they're white. Two little r's means that is recessive. Okay. Capital R means it is homozygous dominant. This is homozygous recessive. And this is heterozygous. You've got two different letters, pink. All right. Here again, you got to know your vocabulary because this is a complete the sentence. Okay. All right. Codominance was like what we had with our calico cat, where you had orange and white and all those colors. This is incomplete dominance because it's neither neither red nor white, it's pink. Okay, so the alleles for this flower show incomplete dominance. So make sure you understand what both of those vocabulary words mean. Because both alleles are partly expressed. Again, it's neither white nor red, it's pink. And they, in heterozygous individuals, okay? All right, anybody? So again, know how, and then while I have this question up, know that you put If they gave you this information, they put one parent on one side, one parent on the other, and you put the information in the box. And this is how they got 100% pink flowers. That's how you read that box. Every box is heterozygous, and every one of those individuals, even though it came from a white flower and a red flower, they're all going to be pink. Okay. All right. This is also an ecology question, except this one, they give you all the information that you need. So this one, you probably could answer without prior knowledge. So ruby-throated hummingbirds are found throughout the North America. They are omnivores, and we are going to talk about food chains and food webs and food pyramids. And we have a list of names: omnivores, carnivores, herbivores, and detrivores. So we're going to look at what all of those things mean. But I think most of you know that an omnivore, because you are an omnivore, eats both plants and animals. Okay, so they are feeding on the nectar of a flower, the pollen in the flower, the tree sap. They can feed on some insects and some spiders. So this is what makes them omnivores. These are your animals, and all the rest of this is plant. All right, so they get all the water they need from the nectar. So that's information. And again, read these paragraphs carefully. They get all the water they need from the nectar. Most nests are in forested areas, citrus groves, marshes, and shrublands. The hummingbirds breed in the spring and the summer, and the females raise 1 to 2 broods during a breeding season. Most hummingbirds migrate to Mexico or Central America in the winter. Many fly nonstop for 20 hours across the Gulf of Mexico. However, and so this is the part you need to pay attention to. However, increasing numbers of hummingbirds have begun to winter north of the Gulf. Researchers have documented that the hummingbird's winter range has expanded northward about 30 miles. All right, so we're looking at our crazy weather right now, and now it's warmer, earlier than it should be. So we have our season of winter. So now a little bit of the southern part of the country is a little warmer than it used to be. So it's starting to look like spring a little earlier. So they can find blooming trees and the flowers and things that they need in order to find nesting areas and find the nectar that they need. And because it's warmer, you're probably going to have some insects that have laid eggs and they're hatching during that time as well. All right, so now you have to look at all of these scenarios and see which task is most important for the ability of hummingbirds to survive the first winter in that environment. All right. All right, so finding stable nesting materials and sites is the most important task for the hummingbird because they must produce offspring. That is important. Okay, so you got to have trees that are blooming in order to be important. So keep that. Don't jump at that, but that's important. Finding sufficient supply of flowering plants, flowering trees, and insects is most important task because they need food to survive. In order to do the first one, you need to have food for energy. So this one takes precedent over this one, even though it's true. You can't build anything. You can't have offspring if you don't have enough energy to make the nest and to mate. So you need to find flowering plant and trees and insects to eat so that you can have enough energy to do everything else. All right, finding bodies of water is the most important task, and to supplement their supply of nectar. Not most important. Not most important. Okay, because they up here it tells you that they get all the water they need from the nectar. So they're not Not important to find a body of water. All right, finding mates to allow humming to lay fertile eggs, etc., etc. Not the first task. Like I said, if you don't have any food and you're too weak, you're not going to need a mate because you don't have any energy to mate and lay eggs. Okay, so finding a place where they can get food, that's most important. Okay. All right. Anybody disagree? Got questions? Stop me if it doesn't make sense. All right. All right. This is something else we haven't covered, but we will. All right. So this thing is called a cladogram. And what it does, this is evolution, and it shows that these invertebrates, again, vocabulary, invertebrates are organisms that do not have an internal skeleton. We are vertebrates. We have an internal skeleton. Okay, so they have diagrammed how they're connected, how they may have evolved, and who's closely related to who. So these are, um, so according to this, and whoever they're closest to on this line um, is is the closest related to them. So I know you may disagree that a worm has anything to do with a spider, but somewhere along the way, they developed legs and body parts. Body segments. So we're going to talk about the fact this one has 3 body segments. So this one, as we go along, they have body segments as well. Okay, but the question says use the drop-down menu to argue the development of 6 legs in invertebrates based on the body structure shown. All right, so over here we've got 8 legs on the spider. This one also has 8 legs, but they seem to have branched off into crickets have let's see, 1, 2, 3, 4. They've got 8 legs. No, that's a tail part. So these are 3 legs, 3 legs, 3 legs, 3 legs. Okay, something 3 pairs of legs. So 6. So somewhere over here during this division, somewhere in this bracket right here, they have developed 6 legs in the vertebrates. So we're looking at segment E. In this segment right here, between here and there, everything from here to there has 6 legs. On the cladogram, because the body structure is present only in organisms at that after that point. So everything after this point, and actually you could argue that D, but everything after D, so let's say it's D, because D splits. Everything after D has 6 legs. Okay. All right. A male and female have a child that has 3 copies of chromosome 18. All right, so that's what we were doing the other day with our karyotype. We looked at the page that had all the chromosomes lined up, and they are numbered. On a karyotype, all the chromosomes are numbered. So on number 18, there's 3 of them. You're only supposed to have 2. Although both parents are unaffected, their doctor claims that the disorder is associated with having an extra chromosome on 18 as a result of a mutation in certain types of cells. Which argument supports this claim? Mutation occurred during crossing over. Again, you need to know what crossing over is. And replicated twice during meiosis. Non-disjunction, and again, the vocabulary word that you need to know, cause an improper separation. And note that mom had 2 and dad had 2, and then they separate, and then they're supposed to go in each cell. So what might have happened is the two that mom had didn't separate and they stayed together. So when dad gave his, you ended up with 3. Okay, so that's how we could get that one. Substitution mutation, that has to do with the genetic lines, but you still need to know vocabulary words. Substitution mutation. Insertion. So this one means that a gene was a gene was changed somewhere along the way. The next one means one was stuck in the line. So remember when we were doing our DNA line and we had the C's and the G's and the T and the A, and sometimes you can take a T out and then it's going to read differently, or we can stick an extra letter in and it'll read differently. So that's what substitution and insertion will do. It'll read differently, but it'll still only make one chromosome. So the thing that caused the extra chromosome to be in the cell is the fact that it did not separate properly during meiosis. Okay? So again, know what these vocabulary words, there's different types of mutations. Know what they are. Vocabulary word. Got it? All right. And then you're going to have the link so you can go back and look at these. And then we're going to look at something else. I'm going to stop in a minute and show you the other page. All right. Students studied Mendel's law of segregation and independent assortment. There you go. Some more vocabulary words. And he created a Punnett square to show how a cross between a homozygous furred mouse and a homozygous furless mouse, which is recessive, the students created the Punnett squares below. All right, so this is the first one, and this is probably this is the second generation. So the first generation produced these, and everybody is heterozygous. And then they took 2 of those, made the second generation, and this is what you got with the second generation. So everybody is heterozygous, but over here, then you get some differences. You got 1 dominant. So you're going to have 1 that is a fur mouse. You're going to have 2 that are heterozygous, but still furred, because for the capital F says that they are that is the characteristic that will show up, and 1 recessive. So you'll have 1 furless mouse. All right, remember that this is possible. These are the 4 possibilities of a cross between these 2 individuals. All right, which question can be answered by using the Punnett square? Can furred mouse produce furless mouse? And that can be answered because these guys are definitely furred, and they did produce a furless mouse in the second generation. All right, how many did the square can tell you how many mice will be produced in generation 1? This just tells you the possibilities of the outcomes. You know, mice, they can have 5, 10, like dogs too. They have, several organisms in the litter, and they could all look the same. Okay, this is just the possibility of what genes they can possibly have. So no, it's not going to tell you how many. Even though there's 4 boxes, the boxes represent probability or possibilities, not the number of offspring. All right, will fewer furless mice survive to adulthood? It's not going to tell you who's going to survive. Again, just tell you the possibility of what can happen if these 2 mate. All right, furred mice are the best organism to use for the investigation. They use both. So how are you going to learn if it's possible to produce furless if you always use furred mice all the time? If you use furred mice all the time, you're going to always get furred mice. So that's not helpful. So we said this one was the best answer because these guys are heterozygous, but they are furred. Okay, so it is possible under this circumstance to produce a furless mouse. Okay. All right. So again, you need to go back and study reading what each one is showing you. All right. So this one is the one I was talking about before, where we had our chart and we had all our nucleotides lined up on the DNA strand. So you have a normal DNA and you have a mutated DNA. And what you're checking is to see if all the letters are the same. And if the letters are in the same order, then you're going to produce the same amino acids. If you produce different amino acids, it's going to be a different protein and it's going to make something else entirely different. All right, a genetic order called Huntington's disease results from the mutation of the HTT gene. The HTT gene provides instructions for making protein called hunting, and the disease is an autosomal dominant disorder. Autosomal means it has to do with, when we were looking at the karyotype, you had 23 of them that dealt with your body and only 2 of them were the sex chromosome. So it is found on one of the 22 other chromosomes. All right, the person needs to only get 1 copy of the defective gene to develop the disorder. Normally, and these are your nucleotides, CAG segment is repeated 10 to 35 times within a gene. In people with the Huntington's disease, CAG segment is repeated more than 35 times, resulting in a protein is cut into smaller fragments that disrupt the normal function of neurons in the brain. It says, move and place labels on the sentence to describe the type of change shown in the model and cellular products. Okay, here we go. They blow it up. So here's those words we were talking about in the last question. This model shows. So what do we have here? And we're looking for CAG. All right, and this one has CAG, CAG, CAG. So this, and it has changed. So this model shows substitution, because right here, we didn't delete anything. We still have the same numbers, but we have substituted, instead of this being normal, instead of having our GTA, now we got another CAG. Instead of having ATTC, we have another CAG. So this is different than that one. Okay, during DNA replication. Okay, so that's what this is. The DNA was copying the information and it didn't copy it right. So therefore, whatever it copied, it totally retrieved different amino acids. All right, so again, I'm going to stop right there and show you the other thing that you can do. So I'm going to stop there. This was question 11. Yeah, we're going to exit. Oops, yeah, exit. All right, because I just want to show you something else. So we're going to come back here to biology. Oops, nope, that's not where I want to be. Okay, come out of that. All right, come back over here. Online tools. Yeah. All right, what you can also do, because you're not going to have me at home with you to talk in your ear, is you can pick the online study guide. And again, you have instructions for that. A lot of instructions. All right, now the difference with this is all that talking I was doing, it's written over here on this side. Okay, so they give you an explanation, clarification, they give you some vocabulary. All of that is explained just so that you can answer this question. Okay. All right, so so did everybody figure out how I got there? I'm going to go back. And so start here. And I want you to spend the rest of class doing this. I want you to look at some on your own. All right, so you're clicking the green arrow like we did before. So you're going to click that with everything. You're going to click tools. You're going to click here. We were doing the practice test, so you can do that. Or you can click on this one that says study guide for students and parents. And then again, you want to click biology. And again, if you're taking one of those other tests, then you can use this for that as well. I'm sure your teacher for that other class is giving you this link as well. I'm going to click on the study guide. All right, and then that'll take you to here. So what I want each of you to do is to start here, read through this question. So read the question. This one is, we've done this one. So this is about cells, prokaryotic cells and eukaryotic cells. You need to know the difference between those. And, and then read through the information that they're giving you. Okay? So in each question is set up like that. They will, and they tell you what the correct answer is. And then they take you to the next question. And this one will also tell you what's going on. Okay? All right, I think you have to scroll for each one. Prokaryotes, viruses, organisms. Okay. See, we're going to spend time with this. After the test, we're going to do a biome project, role of species. So just reading through this would be very helpful because this covers a lot of the information that we have not done yet. Okay. So read through that. All right. So again, I want each of you to spend the rest of the time looking at this on your own. And again, tomorrow we'll do this, and let's see, Friday we'll do some more. Okay? But spend some time doing this on your own. All right, checking on my testing people. Did you finish, Andre? Because I know you only had to finish. All right. Alicia, did you do the test? I'm looking at your screen and it says begin. That means you have not begun. Oh, that's the bell ringer. Okay. Never mind. Don't worry about that one. You wanted to be doing the benchmark. Did you do the benchmark? You got to finish because he's going to close it, and then you'll have to finish it again another day because he's only going to open it when I ask him to open it. And you haven't started because it says 40 questions. People, please use class time for classwork. Don't log into class and then do something else, especially for things like this, because this is a timed situation. He is not going to open this back up unless I ask him to next class. All right, so the rest of y'all, like I said, use class time for classwork. We have to pass this test. We have to pass this test. We have to pass this test. So practice makes perfect. The way to get better is to practice. And again, you need to familiarize yourself with the way they ask questions because they really want you to think. Okay, they want you to know what you're talking about. You don't get to take the book. You don't get to scroll and cheat and look up Google anything. So you got to go in there knowing some things. So please use this time over the break to study. And I hope you were following along with me. Okay. Actually, let me do that for everybody. Because I did that with the last class. Because that was something that everybody kind of had a moment with. All right, so if you haven't turned in your karyotyping worksheet, I am going to go over those answers right now. I know I told you to work on that, but if you need help with the karyotyping worksheet, I'm about to go over those answers right now, so consider this review as well. All right. Remember, the whole picture is a karyotype. Karyotype simply means, and then chromosomes don't look like this in the cell. They're all scattered all over the place. But what they do with the karyotype is they sort them out and they put all the ones that are alike together. And so this is a mapping of all the chromosomes that are in that particular cell. These are human cells. Human cells in a normal person, I'm answering number 2, is 46. Now, normally it said total chromosomes. The picture is showing 23 pairs, okay? But the question said total chromosomes. Again, read each thing carefully and don't, because you could just say, oh, I look over here, I see 23. 23 is the answer. That's not the answer. They said total chromosomes. 23 times 10 is 2 of them. So it's 46. All right, so what you had to do is that autosomal chromosomes, those are all the chromosomes except the sex chromosomes. All right, so you could have just put one big circle around everything except that last 2. Okay, so some of you put circles around each one, and that's fine. But you could have just put a circle around a 1 through 22, one big circle. Put a box around the sex chromosomes. Those are always the ones on the 23. So this one is X and Y. So that tells you that this person is a male. Okay, and if you weren't sure, you go back to in Canvas. Now I wanted to point this one out too. Chromosomes are not this neat. So this picture is very neat and structured. Chromosomes aren't straight little sticks like that. Okay, they are curly like the ones that you just saw in the other picture. But this one is showing you again 1 through 22. Those are your autosomes, and then 23 is going to either or. You're going to have a long one for X and a short one for Y, or you'll have 2 long ones being 2 X's. Y is a boy, XX is a girl. Okay. All right. Coming back to the worksheet. I got like 2 minutes. Abinay, which question specifically, because I may not get to it in the next 2 minutes. So I've answered all the first one. It said put a star around a homologous pair. You could have put the star anywhere because all of these are homologous pairs. Homologous pairs means that they are 2 of the same kind. Notice they're all different sizes. So the 2 long ones are together, the short ones are together. If they have the same information on it, they're together. So any of these are homologous pairs, even this, except for the X and the Y. All right, so that's that one. It says looking at the karyotype to the right. That's this next set right here. Do you see any abnormalities? You need number 14. Okay, so you're good. You're good on all of that. Let me go this room real quick. So the abnormality here is on this the sex chromosomes. Okay, there's 3 of them. There should only be 2. So you go down here to the chart, and when you have 3 of them, it's triple X disease. A lot of you got this one wrong. Triple X disease because there were 3 X chromosomes. All right, I'm gonna skip down. She said, what type of mutation is number 14? So you come down here to this chart, and then they'll have a chart like this for you on the test as well. So 21 is Down syndrome. Trisomy 21 is called Down syndrome. Okay. All right. This one here, real quick, we're looking at the disorder in figure A. So this is figure A, and we're looking at all the pictures. They've got, it says X and Y, but there's nothing over here with the Y. So we jump back up here, and we see this one has 1 X, no Y. That's Turner syndrome. So this is Turner syndrome. It was should have been a male. Okay. 1 X, no Y, no other X. All right. Identify the disorder. This one has over here in the sex chromosome, they've got an X and 2 Ys. So we come back up here, look for X and 2 Ys. That's Jacobs syndrome. And that's going to be male. Over here, your problem is on 13. You've got 3 of them on number 13. Come back up to your chart. Trisomy 13, Patau syndrome. And this one is a female. They've got 2 X chromosomes. All right, this one over here was right here. You've got 3 of them on 2021. Trisomy 21, Down syndrome. Okay, and then this one, you've got 2 X's and a Y. So it's a male, and 2 X's and a Y is Klinefelter syndrome. Okay, so that's how you read these. You have to scan through each of them, and remember that you're only supposed to have 2, but you do need to have 2. So if you're missing one, that's a defect. Okay. All right, folks, it's time to go. So I'll see you tomorrow. And any other questions that you have. And again, this we'll be reviewing from now till test time. So if there's a topic that you want me to discuss with you that totally made you scratch your head, let me know. Otherwise, I'll just keep going over the review questions. Have a great day. Enjoy your lunch. Bye-bye.